Good morning students, we will continue the topic biological oxidation computation number 6.6 and today's topic is uncouplers. Uncouplers are the compounds which allow the oxidation to proceed in the electron transport chain, but the energy release during this transfer of electron is not utilized for synthesis of ATP. That means phosphorylation does not takes place and that energy is released in the form of heat. Such compounds are called as uncouplers. How this occurs? This is achieved by removal of proton gradient by providing alternate pathway for the protons. In the last lecture I have told you that when electron transported through the FO-F1 complex, then only ATP, when protons transferred through FO-F1 complex, then only ATP is synthesized. But these uncouplers made this inner mitochondrial membrane permeable for protons, and hence protons directly passes from intermembranous space to matrix bypassing the FO-F1 complex. When proton gradient is dissipated, or when the proton does not passes through FO-F1 subunit and when they passes directly through the inner mitochondrial membrane in the presence of uncoupler, ATP is not synthesized. So these are the examples of uncouplers. Few are physiological, like thyroxine in high doses. Thyroxine means thyroid hormone. Thermogenin, it is a brown fat present in our body. Other uncouplers are 2,4-dinitrophenol, 2,4-dinitrocresol, chlorocarbonyl, cyanide, phenyl, hydrazine. Along with uncouplers, some ionophores also act as a, or they dissipate the proton gradient by allowing the potassium ion travel through the inner mitochondrial membrane. For example, see here again the proton gradient is there across the inner mitochondrial membrane. Here in the intermembranous space more protons are present, on the matrix less protons are present. That means positive charges are more in the intermembranous space and less positive charges are present in the matrix. Now the ionophores allow the potassium ion to travel through the inner mitochondrial membrane from intermembranous space to matrix. When potassium travels in this direction, the positive charges on the intermembranous space decreases and they increases on the matrix side. When the positive charges is balanced on both sides of the inner mitochondrial membrane, the movement of proton across the FO-F1 subunit does not occurs, and in this way the ATP synthesis does not take place. The examples of these ionophores are valinomycin and gramicidin. This is about uncouplers of oxidative phosphorylation. Thank you very much.